right? Here, what production, what type of the seeds are going to be produced? Intermediate size. Intermediate size is present in the parent? No. Parents are what? Dominants only. Means either big or small only, right? So if starch grain size is considered as the phenotype, so alleles show incomplete dominance. You got it or not? So in case of heterozygous condition of the parents, so the seeds are production of the seeds are intermediate size. Intermediate, it is not presented in the parents. Parents are either big size or small size only. You got it or not? Right? So for incomplete dominance, this is also one of the example, right? Starch P of the starch production gene also one of the example, right? So here we are discussing regarding about Pliotropy. Okay, so you can consider for Pliotropy example for starch P only. So here see, again I am revising. Generally one gene controls one phenotype, but sometimes one gene may control more than one phenotype. One gene controls one phenotype, and but sometimes one gene may controls more than one phenotype also. That is called, that scenario is called Pleiotropy. And the genes that control more than one phenotype are called pleiotropic genes, right? Okay, a pleiotropic gene controls a major phenotypic trait, and along with that it influences some other phenotypic trait also. The gene has two responsibility, one is major and another other also. So what other major phenotypic trait, and another one also it controls another phenotypic trait also, right? Okay, so what is the best example for pleiotropy? And starch synthesis in pea seeds. The synthesis of synthesis of starch in pea seeds, it is controlled by one gene, which also influences shape of the seeds, right? So the major controlling is synthesis of starch in pea plant, and the another controlling is the shape of a seed, right? It has two alleles. So what is the type of two alleles? There is two alleles, capital B and small b, right? Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In case capital B is presented, the size of the starch is large, right? And starch synthesis also effectively, whenever starch synthesis is effective, then only the size is increased, obviously, right? In contrast, small b homozygotes have lesser efficiency in starch synthesis and produced smaller starch grains, right? In case of small b is presented homozygotic condition, right? This is recessive and lesser starch synthesis efficiency, and production of the size of the seeds also small. After maturation of these seeds, capital B seeds are round, and small b seeds are wrinkled. So round and wrinkled is responsible for by that gene only, that same gene only, and starch synthesis production also by that same gene only. So that only it is example for pleiotropy. And heterozygotes produced round seeds, capital B dominant allele. So heterozygotes always produce round seeds only. So capital B is the dominant allele. Here why? Because of capital B is dominant allele, so that only capital B small b present also heterozygote condition, that is exhibits a round shape only. The starch grain produced are of intermediate size in capital B small b size. In seeds, in case of heterozygous condition is presented, capital B small b. So what type of the seed size, big or small? So here intermediate size, neither big nor small, so that only these type of the alleles exhibits incomplete dominance. So here mixing of the character, blending of the characters takes place. So if starch grain size is considered as phenotype, the allele shows incomplete dominance. Why incomplete dominance? Because those are not similar with the parents, right? Okay, so next one, what is the examples, another examples of pleiotropy in humans? So till now we discussed pleiotropy examples in plant, pea plant. So in human pleiotropy examples, in humans pleiotropy examples are what? Phenylketonuria, PKU, and sickle cell anemia. Phenylketonuria, PKU, and sickle cell anemia. These two are different types of genetic disorders, and these two disorders, regarding about these two disorders, we will discuss later topics, next topics. So phenylketonuria and sickle cell anemia, these two are examples for pleiotropy in the humans, right? Phenylketonuria and sickle cell anemia, these two are the different types of the disorders related to the chromosomes and genes, and these are also examples for pleiotropy in the humans, right? Up to this clear? Hope all of you understood up to this topic, right? Go through the NCERT line and video classes also, and in next topic we will start chromosomal theory of inheritance, right? Okay, hope all of you preparing whatever the questions given related to the reproductive health 1 mark questions. So I will check after 1 or 2 days. I'm giving 1 or 2 days time. You can take time and prepare the answers, right? Okay. So, and one more point, hope all of you preparing this weekend exam also, NEET also, and whatever the last weekend exam, I observe the result, but not very satisfied. Some of the members are done well, but most of the members are not up to the mark. So whatever the concepts, just go through the NCERT lines, right? Any doubts you can ask through the messages, and you can call me directly, right? So already we discussed it so many times, the human physiology chapters are most important, very important in the NEET point of view, in the objective point of view. You can be thorough with the human physiology chapters, all the human physiology chapters, right? And this is the best time for preparation and best time for the doing of objectives also, right? Thank you very much. Bye bye.